Okay, Shafi, today we shall discuss on an interesting topic, cell junctions, cell adhesion, and extracellular matrix. In our uh, previous discussion, we talked about cell cytoskeleton and associated proteins and their function in cell structure, cell shape, as well as cell division. Today, the cell junction, cell adhesion, and the extracellular matrix. These are very important and fundamental topics in cell biology, cell and molecular biology. Cell junctions. First, let us know about this. Cell junction is a type of structure that exists within the tissues of some multicellular organisms such as animals, even in plants. Cell junctions consist of multi-protein complexes like cell cytoskeleton. Cell junctions consist of multi-protein complexes that provide contact between neighboring cells and between a cell and the extracellular matrix. Cell junctions are especially abundant in epithelial tissues in the animal. Cell junctions are especially important in enabling communication between neighboring cells via specialized proteins called communicating junctions. That means communicating junctions involved in communication between two neighboring cells. Cell junctions are also important in reducing stress placed upon the cell. So what we learned from this slide, cell junctions, they are actually multi-protein complexes and they are involved in stress tolerance of cell. That means reduce the stresses, external stresses, and most importantly, they are involved in communication between neighboring cells. Here you can see a nice illustration. Uh, Here you can see there are some junctions. Epithelial tissue, basal lamina, and connective tissue. There are some other protein you see, collagen. So there are here you see the epithelial tissue involved in reducing the mechanical stresses. Mechanical stresses are transmitted from cell to cell by cytoskeletal filaments anchored to the cell matrix or cell to cell adhesion site. Here you can see these filaments. Filaments are, already cytoskeleton. Extracellular matrix directly bears mechanical stresses of the tension and compression. These are the extracellular matrix. So there are two main ways in which animal cells are bound together: connective tissues and epithelial tissues. Connective tissues and epithelial tissues. Here you see connective tissues, and here is the epithelial tissues. So four functional types of cell junction we already been known. So cell junctions can be classified into four functional groups. The first one, occluding junction, seal cells together in an epithelial in a way that prevents even small molecules from leaking from one side to the sheet to other side. Anchoring junction, mechanically attach cells and their cytoskeleton to their neighbors and to the extracellular matrix. So occluding junction, anchoring junction. Third, channel forming junctions create passageways linking the cytoplasms of adjacent cells. Fourth, signal relaying junctions allow signals to be relayed from cell to cell across their plasma membrane at site of cell-to-cell contact. So signal relaying junction, signal transduction is very important, and here signal relaying junctions are involved. Here a very beautiful illustration. You can now get more understanding about four types of cell junctions. Here you see anchoring junction. Anchor, you see, anchoring. This junction anchors. These are protein complexes, okay, anchoring. You see, cell to cell anchor, uh, like junction, station or like that. Occluding junctions. Here you see occluding junctions. Channel forming junctions. This protein and this protein, they are involved in channel forming so that ion can pass through from or molecules from one cell to another or from external source to the interior of the cell. Signal relaying junctions. These are signal relaying junctions. So signal pass from one cell to another. So these are very beautiful. And if you go through molecular biology of the cell, you can find details about these four types of junctions. So this is a table, a functional classification of cell junction, what functions they are doing. For example, anchoring junctions, occluding junctions, channel forming junctions, signal relaying junctions. Here in this table, it is listed the major functions of each type of junction. Anchoring junction, actin filament attachment sites. That means cell to cell function, adherence junctions, cell matrix junctions, actin linked cell matrix adhesion, intermediate filament as attachment site. That means actin and intermediate filament, they are cytoskeletal proteins. So cell to cell junctions, in case of intermediate filaments, they are called desmosomes. They are also protein. Cell matrix junction, hemidesmosomes. That means adherence junction, actin-linked cell matrix junction, adhesions, desmosomes, and hemidesmosomes. These are involved in anchoring of the cell. Occluding junctions, tight junctions in the vertebrates, septate junctions in the invertebrate, and channel forming junctions are two types. Gap junctions in animals, plasmodesmata, you possibly heard about it, plasmodesmata. They are involved in channel forming activity in the plants. So gap junctions are present in the animals and plasmodesmata in plants, and they are both channel forming junctions. Signal relaying junctions, they are chemical synapses like in the nervous system, immunological synapses in the immune system, transmembrane ligand receptor cell-to-cell signaling contacts, delta notch, afrin, like that. Anchoring, occluding, and channel forming junctions can all have signaling functions in addition to their structural roles. That means signaling functions are also present in other junctions. Summary of the various types of cell junctions can be seen by this excellent illustration. Here you see the apical part. This is the basal part. The cell. Here you see occluding junction. Occluding junction, tight junction seals gap between epithelial cells, between two cells. Okay, and adherence junction connects actin filaments bundle to the cell with the next one. These are the actin filaments. So these junctions attach uh, the actin filaments, adherence. So one cell can adhere to others. Cell to cell anchoring junctions, cell to cell anchoring. You see, these are the anchoring. So these proteins, they are anchored from one cell to another so that they can stay together. These are the channel forming junctions. So this is one cell, this is another cell, this is another cell. So channel that allows ion and molecules to pass through from one cell to another. Cell matrix anchoring junction. Here you see cell matrix, uh, uh, how they are connected by using special protein. Gap junctions allow the passage of small water-soluble molecules from cell to cell. For example, glucose can pass through the gap junction. So these are very interesting. Here you see uh, hemidesmosome anchors, uh, intermediate filaments in a cell to extracellular matrix. Actin-linked cell matrix adhesion anchors actin filaments in cell to extracellular matrix. So uh, all the cell junctions are shown in the simple, simplest diagram, which I took from Molecular Biology of the Cell book. Tight junctions in occluding function, tight junction has occluding function. For example, they function as barriers to the diffusion of some membrane proteins and lipids between apical and basolateral domains of the plasma membrane. Here you can see sodium-driven glucose import, how they are involved. You see, glucose can pass through apical surface, lumen of gut. Do you know, in our intestine, these are the lumen, and here glucose can we take lots of carbohydrates, so glucose after digestion they can pass through this type of you know the tight junction they can pass. Similarly, there are lots of other junctions, tight junction, plasma membrane of the adjacent cells. So here you can see two cells and intracellular spaces, passive glucose, carrier protein. Sometimes, all junctions are protein, but sometimes carrier proteins are also involved. So finally, after uh, entering into the cell, glucose finally moved to the blood. But in the villi cell, in the intestine, they are, entered into the cellular system from the intestine through this tight junction. So gradually moves to the blood, because in the blood cell, glucose is burned in the mitochondria to release the ATP. So tight junctions seal neighboring cells together so that if a low molecular weight tracer is added to one side of the epithelium, it will generally not pass beyond the tight junction. Here you can see tight junctions. So the cell molecule cannot move up from one cell to another, and there are some gatekeeping system, and these are the electron microscopic image, and these are the drawings so that you can easily understand. So main feature of the tight junction uh, is the tight junction may serve as a regulatory center to help in coordinating multiple cell processes. Epithelial cell can transiently alter their tight junction to permit an increased flow of solutes and water through breaches in the junction barriers. Such paracellular transport is monosaccharides, especially important in the absorption of amino acids and monosaccharides. from the lumen of the intestine, because when we take protein food containing rich in protein or carbohydrate, ultimately in the intestine they are digested, and finally amino acids, monosaccharides, these small molecules are transport, which is called paracellular transport, and finally moves to the blood. So when tight junction are visualized by freeze fracture electron microscopy, here freeze fracture electron microscopy, they show the tight junction very nicely. Here you see. So it is not simply drawing, but practically scientists did freeze fracture electron microscopy to visualize the tight junction, how sealing strands networks are present. In conventional electron micrograph, the outer leaflets of the two interacting plasma membrane are seen in the tightly opposed where sealing the strand are present. Figure C, you can see here. So uh, these are all cell biological study, uh, well established, and here you can see more clear molecular biology of the cell, fourth edition. This illustration I took from there. Each tight junction sealings as a strand is composed of a long row. Here you see interacting plasma membrane. This is a plasma membrane. This is a plasma membrane. And here you can see sealing strand of tight junction. You see they are basically protein, and how beneath the plasma membrane to cell, how tightly sealing strands of tight junction protein they are working. Here you can see in B, cell 1, cell 2. Here you see how they are working. So uh, uh, clouding and occluding. Clouding, these are clouding and these are occluding. How they are involved in sealing the two cells, neighboring cells, and clouding and occluding, they are different protein and they are called tight junction proteins. So very interesting. So cloudin and occludin are the proteins involved in tight junction. Cloudin, which are essential for tight junction formation and the function, and differ in different tight junctions. A specific cloudin found in kidney epithelial cells, for example, is required magnesium ion to be resorbed from the urine into the blood. A mutation in the gene encoding the cloudin results in excessive loss of magnesium in the urine. Occludin, they are very crucial, you see. Occludin, the function of which is uncertain. Still, scientists don't know the real function of occludin. But cloudin and occludins associated with intracellular peripheral membrane proteins called ZO protein. A tight junction is also known as zonula occludens. A tight junction is also known as a zonula occludens, which anchor the strand to the actin cytoskeleton. So anchoring junction connect the cytoskeleton of the cell either to the cytoskeleton of its neighbor and to the extracellular matrix. Here you see these are the cytoskeletal filaments. Okay, and anchoring junction are here. You see cytoskeleton here in one cell and another cell. They are connected by the anchoring junction. So cytoskeleton skeletal proteins are anchored into the anchoring junctions. Very nicely here it is shown. Um, Anchoring junctions are widely distributed in animal tissues, most abundant in tissues that are subjected to sharp mechanical stress, such as heart, muscle, epidermis. Here you see intracellular anchor proteins. Here you see intracellular anchor proteins, cell 1 and cell 2. How you see transmembrane adhesion proteins are involved in connecting the cytoskeletal filament. This is a cytoskeletal filament. This is a cytoskeletal filament. Blue are the cytoskeletal filament, but they are the this is the plasma membrane. So how intracellular anchor protein, these are the intracellular anchor protein, connect one cell to another to anchor each other. So it is shown here. And there are some transmembrane adhesion protein. These are the transmembrane. You see, this is a membrane of one cell, this is membrane of other cells. So they are transmembrane adhesion to adhere of one cell to another. So there are two types of proteins are involved in anchoring junction. One is cadherin, another is integrins. Cadherin means cell to cell, uh, uh, anchoring, and integrins cell matrix. That means or with the cell matrix. Here you see cell to cell. So cadherin and integrins, they are involved. They are composed of two main classes of proteins, as I mentioned. Intracellular anchor proteins from a distinct plaque on the cytoplasmic face of the plasma membrane and connect the junction complex to either actin filaments or intermediate filaments. And transmembrane adhesion protein have the cytoplasmic tail and binds to one or more intracellular anchor proteins and extracellular domain that interacts with either the extracellular matrix or the extracellular domain of specific transmembrane adhesion proteins or another cell. In addition to anchor proteins, adhesion proteins may anchoring junction contain intracellular signaling proteins that enable the junctions to signal to the interior. That means external signal can be passed to the interior. So anchoring junctions occur in two functionally different forms. Adherent junctions or desmosomes hold cells together and form the transmembrane adhesion proteins that belong to the cadherin family. So cadherin family is a large family of protein that are involved in adherence junction. Focal adhesion and hemidesmosome binds the cells to the extracellular matrix are formed transmembrane adhesion proteins called integrins. That means extracellular anchoring or adhesion to the extracellular matrix, the anchoring junction uses integrin family of proteins for this purpose. On the other hand, cell to cell one cell hold with others, the proteins involved in anchoring junction is called cadherin family. I showed you in a previous slide. So cadherins and desmosomes, you can see here very clearly. Cell to cell connections are mediated by cadherins. Cell to cell, connections. Here you see, these are the here is the plasma membrane, here is the plasma membrane, cell 1, cell 2. These proteins are called cadherin dimers. So cadherin, here is the, you see actin filaments, reds are actin filaments, and these are the anchor protein. So how they are connected, and these receptor, they are called receptors also because they are anchored into the plasma membrane. These receptors extend out from the cell, binding to other cadherins. Here you see cadherins from one cell and cadherin from other cells. They are bound each other and tightly adhere the cells. Cadherin participate in adherence junctions. Here you can see how cadherin participates in adherence junction. For example, adhesion. Here you can see actin filaments inside the microvillus lumen. This is the intestinal lumen, microvilli extending from the apical. So here you see tight junction, cadherins here, uh, uh, how cadherins adheres the bundle of actin filaments, and two cells are how they are tightly uh, uh, attaches. So they surround the cell, forming a belt-like structure. Desmosomes, cadherins can also form localized spot connections. Localized spot correction. Here you see cadherin family proteins. Here is one cell plasma membrane, another cell plasma membrane. And here you see keratin filaments anchored to the cytoplasm. And here also a different platform of anchoring protein. But how they are, in the uh, how they are connected to each other. Here is more elaborated. Desmoglein and desmocholine, a different type of protein, but they are working with the intermediate filament, that is cytoskeleton, how they are connected. So lots of protein, for example, placoglobin, plaquing, scientists already discovered lots of things. So here is cell 1, cell 2, here is the plasma membrane 1, plasma membrane 2, how they are connected, like very nicely, engineering structure and amazing in the living cell. So anchoring junctions formed by integrins bind cells to extracellular matrix, focal adhesion, and hemidesmosomes. The transmembrane adhesion proteins in the cell matrix junctions are integrins. Already we discussed a large family of proteins distinct from the cadherins. Focal adhesion enables cells to get hold on the extracellular matrix. That means integrins works there through integrins that link intracellularly to actin filaments. Hemidesmosomes connect the basal surface of an epithelial cells to the underlying basal lamina. Here, cell-to-cell extracellular matrix attachment is seen, and the upper A and B, you can see they are, microscopic images, but lower illustration is very clear. Extracellular matrix protein, and then you see integrin subunits. These are alpha and beta, different structural variation in the protein plasma membrane of one cell. So this is extracellular space, and how another it enters into the another cell cytoplasm. That means alpha actin, taurine, or filamin, vinculin, lots of actin, they are participates and work together. So focal adhesion use microfilament, and hemidesmosomes use intermediate filament. In case of actin, cell cytoskeleton, tubulin, microfilament, and intermediate filament, three types of protein. So they are involved in holding the cell and anchoring the cell, attach the cell with one another. So lots of information have been known, and we can summarize the anchoring junctions like this way: transmembrane adhesion protein, extracellular ligand, and intracellular cytoskeletal attachment, intracellular anchor protein. So these are the junction protein: adherent junction, desmosomes, actin linked cell matrix adhesion, hemidesmosomes. That means two type of cell to cell anchoring, and cell to matrix anchoring. So these two major type of function is done by the junction anchoring proteins. And here is the or different kind of proteins they are involved already been discovered in case of transmembrane adhesion protein, extracellular ligand, intracellular cytoskeletal attachment, for example, actin filaments or intermediate filaments, or intracellular anchor proteins. Lots of intracellular anchor proteins have already been discovered. So we showed through the images or illustration. So if you take this one with the illustration, then you can get the clear uh, information. So another junction is called gap junction. What is that? Gap junction allows cells to exchange electrical or chemical signal. So electrical and chemical signals are mediated through the gap junctions. And gap junctions, here you can see the image, how much gap? A gap of 2 to 4 nanometer, incredibly small. So to connection in the register forming open channel between the adjacent cell, and they are involved there, channel 1.5 nanometer or like that. So scientists already through using transmission electron microscopy they discovered. So there are lots of proteins involved, connexin, connexons, intracellular channels. They are involved in gap junction. And they can be, homomeric or heteromeric, different kind of, maybe involved. So summary, tight junction, uh, adherent junction, desmosome, gap junction, hemidesmosomes. If we can summarize, tight junction seals neighboring cells together in an epithelial sheet to prevent leakage of molecules between them. This is the main function of the tight junction. Adherent junction joins an actin bundle in one cell to a similar bundle in the neighboring cell. Cytoskeleton of two cells are connected by the adhered to the adherence junction. Desmosome joins the intermediate filament, not the actin filament. Intermediate filament in one cell to those in the neighbor. Gap junction allows the passage of small water-soluble ions and molecules. Hemidesmosomes anchors intermediate filaments in a cell to the basal lamina. So these are very simplest way and major function uh, uh, are shown in this table in summary. So these are in short cell junctions, cell adhesion, and the extracellular matrix. So these are the fascinating area of research because cell-to-cell communication, in an organ, lots of cells are there and number of cells are pre-programmed. How many cells in an organ, for example, in your finger, how many cells would be accommodated, it is pre-programmed. So after certain period of time, they are, aged and then they are replaced. And every cell are connected to each other and also they are connected to the extracellular matrix by using different junctions. And basically in cell biology, the more you learn, the more you can understand how how much diversity in the proteins are in our cell, and proteins are actually the real workers for all cellular activities that we can see. We are different, and all diversity we can see, they are due to the presence of different proteins. Obviously, all are regulated by the function of or expression of particular genes. Thank you very much. Any question?
Yes, sir. Signal transduction is much more complex in case of animals.
Signal transduction pathway in the animals hugely been studied, and these different cell junctions, cell adhesion, extracellular matrix. These are the fundamental information will help you when we discuss about different signal transduction pathway. For example, ion channel or G protein couple receptor mediated pathway. So a signal can be chemical signal, signal can be physical signal, for example, temperature, high temperature or cold, or even, other biotic and abiotic signal. Signal can be a living organism also. So signal transduction obviously in animal very complex, but also complex in the other eukaryotes. But obviously in human and other mammalian system, they are more complex than signaling pathway in the unicellular organism like bacteria. Uh, a relatively single cellular, unicellular organism, signal transduction pathway are uh, simpler compared to the complex organism like animal. Any question?
No, sir.
Then thank you so much. And next week I shall, maybe in Saturday, if I arrange another lecture, is it okay with you?
Yes, sir.
Okay, then thank you so much. And please enjoy. You have this lecture, right, with you?
No, no, sir.
I send you through WeTransfer all the files.
Oh, okay, sir. Then I'll check, sir.
Please download all the files and you can get all the lectures.
Okay,
But
sir.
the sequence of the lectures may be different because I'm changing depending on the, to make a coherence in the lecture so that one is connected to another so that you can follow nicely. Thank you so much and good night.